Today I will present about the shotgun metagenomic approach for studying the microbiome in shrimp aquaculture in Vietnam. And this project was funded by the International Development Research Center of Canada. Okay. So aquaculture play a vital role in many countries, especially the developing countries in Southeast Asia like Vietnam, Thailand, Indonesia, and also Philippines. And one type of aquaculture practice, the wildlife shrimp, the, the wildlife shrimp becoming the main crustacean in the international worldwide. And with an output of 6.8 billion tons in 2022, the wildlife shrimp becoming the most species producing. Vietnam, with a long coastline and tropical climate, is an ideally condition for developing the industry aquaculture. And the primary export of Vietnam is shrimp farm, shrimp fish, and also the tuna. And nowadays, Vietnam is leading is one of the top world country in the producing and exporting shrimp. And in 2024, Vietnam managed about 750,000 hectares with an output of 1.3 million tons. And the wildlife shrimp will accounting for 950,000 tons of this total. There are two types of shrimp farming in Vietnam, the extensive and the intensively. While the intensively occurs in a natural pond with a low stock density and minimal input, the intensively using with the high stock density and advanced technology to maximize the production of in the control and environmental. So like any other developing country with the intensively culture, we're facing a big challenge. With a high stock density and an excessive breeding, there is an accumulation of the harmful substances in the shrimp pond. And this situation leading to not only harmful the environment quality, but also raise a high risk of disease outbreak. So to address this challenge effectively, there is a very essential to understand the role of the environmental, especially the microbiome, how the microbiome of the environmental play in the maintaining the shrimp health and also preventing the disease. So for this purpose, in the preliminary study, we employing a shotgun metagenomic approach to characterize the microbiome community and also the antibiotic resistant profile in the shrimp rearing pond in Vietnam. The study was take place in two provinces, one in the north central of Vietnam and the other in the southern of Vietnam. And at each province, we will come to two types of shrimp farming, the extensively cultured pond and the intensively cultured pond. And at each pond, we will two types of sample were collected, the bottom sediment or feces and the water culture. The sample were collected in triplicate, and after DNA extraction, the triplicate were pulled together for shotgun sequencing. So, and we will investigate the taxonomic classification and also the antimicrobial resistance gene in the sample. So here is information of the sample I will discuss today. The sample were highlighted in red is will come from the intensively pond. So across on the eight sample, there is about 50 gigabyte data were obtained with an average of 23 million reads per sample. And as you can see here, there is a lot of read across sample were unclassified. So about an average of 70% of the read per sample is not classified at any taxonomic level due to the absence of the database. So contrary to the human microbiome, there is still a lot of things in the environmental that we didn't know yet. And the taxonomic classification is mainly the abundance of the bacteria. For the taxonomic classification, we see that the actinomycetota and the pseudomonadota is across all of the S sample. However, when compared to type of the shrimp farming, you can see that the cyanobacteriota is more abundant at the intensive one, when the phylum, the Bacteroidota, is more abundant in the intensive one. And similar results were observed at the genus level, while there is a lot of Xanthomonas or Pseudomonas is abundant at the intensive one. The intensive one is highly abundant with the Rugeria. We also compare the alpha diversity, and even with the sample from the extensively had a higher number of species observed and also higher the Shannon index, but there is still no difference between two types of shrimp farming. For the beta diversity analysis, we are computing the PCOA, the principal coordinate analysis, with the birecurrent dissimilarity, and at each point here is represent the sample, and the distance between these dots will reflect how the similarity or the difference in the each other. And we found that sample come from the same source, like they come from the bottom sediment, or come from the water, or come from the feces, is more likely similar than those merely co-located. And and the pairwise permanova indicated that there is a significant difference between the extensively and the intensively one. Following the taxonomic classification, we also want to investigate the functional annotation of microbiome community with profiling the antimicrobial resistant gene in a sample. And so it's really clear to see that the intensively had the higher number of the AMR gene, and also had the higher number of Thai AMR gene compared to the extensively one, and there is a significant difference between two types of sample. And with the largest share of aquatic animal production, Vietnam is one of the top 4 countries had the highest antibiotic consumption. So the ministry had the list of 24 antibiotics which are prohibited in aquaculture. So to assess the potential violation of the antibiotic regulation, we will compare the AMR gene profile with the list of these antibiotics. So you can see here that there is a higher number of AMR genes that are related to the resistant to the aminoglycoside, macrolide, phenol, or tetracycline. And the higher number of these AMR genes will reflect that because it's due to the widely spread use in the aquaculture. Furthermore, among the resistant genes detected, we found that in the intensively one, there is a higher number of AMR genes related to the fluoroquinolone resistant, which are the fluoroquinolone which are invented in the Vietnam, which are banned in the aquatic culture using in Vietnam. So there is a raise and high awareness and also a strictly control in the usage of the antibiotic usage in the aquaculture. For this study, we conclude that there is a significant difference in the diversity between the extensively and the intensively cultured point. A higher number of AMR genes were detected in the intensive point, including the antibiotics that are prohibited, raising an awareness of the antibiotic usage in the farm. And we want to notice that the shotgun metagenomic approach can be further used for developing management strategy to sustain the stream health and can contribute to the improved ecological management of aquatic ecosystem. Thank you for your listening.